You have genetic engineering. Under this, you can have cloning. You can have biotechnology. You can have RDT. That is recombinant DNA technology. That's the one you're talking about. Recombinant DNA technology. Now, recombinant DNA technology can also be used under biotechnology. Because recombinant DNA technology is simply the technology during which we cut genes from, say, one organism and insert it inside an unrelated organism. So we first have a normal combination of genes existing naturally. If we cut it out and put it somewhere else, what are we doing? There was first a combination. The moment we cut it out and put it, what are we doing? We are recombining. That's where the name comes from. Recombinant DNA technology. It's like I take here, I paste that side to combine in a new set somewhere. I'm recombining now. Now, before I explain to you the method, I want to make you understand why we went to such extent of applying such technologies like genetic engineering in general. First off, take for instance, I have a certain plant, okay, and that plant is a tomato crop. I'm a farmer, I grow tomatoes. But somehow, there are some insects. These insects are pests. What do? They attack and destroy my tomato. So when they come to my tomato, they will possibly kill the tomato. Okay? Because they are sucking the sap out of the tomato and all of that. Now I realize that these same insects, once in a while, they also try to feed on, draw it like that. Okay, these are the in Mamelodi. They also try to feed on pigs. This is a pig, the type you find in Mamelodi. So now when these insects feed on these pigs, I notice something. It doesn't cause any harm to the pig, but rather the insects themselves, they die. Okay? The insects die. After a meal on the pig, they die. So I ask myself, why are these insects dying when they feed on pigs? Even if I can't answer that question, at least, no matter how uneducated I could be, I'm able to guess that inside that pig there is what? There is something, isn't it? Something that kills the insects. That alone is enough for me to use it as a start-off point. I need to be able to say there is something. So that something is what now I'll be interested in. Okay? That is what I'm interested in. Something. Now, would you wish that your plants should also have that something? Would you wish? You would wish for that, right? Good. Now, let's leave this picture where it is. We'll come back to it. I have 2 cows. Cow A produces 20 liters of milk per day. Cow B produces 60 liters of milk per day. I'm a dairy farmer. Which cow would you prefer to keep between these two, A or B? B. Of course, Africans prefer quantity over quality. Okay, so it's good. So, because because it produces a lot of milk, I have more products to sell. B has that milk production. I want us to understand. Milk production is what? Is a characteristic. Do you realize that? Which is also a what? A trait, isn't it? And that trait here is high milk yield. That is a trait that this cow has. This cow has a trait of high milk yield. In the meantime, it is a characteristic. Now, you said you prefer to keep cow B, correct? And would you wish to have many of such cows? Yes. How would you go about having many of them?
By finding the something in cow B that
Forget about that part. This is the problem. When we go into genetic engineering, we start thinking only in terms of genetic engineering. We are locked up there. Think naturally. What would you do? What would you think of? You have a cow in your farm that's producing a lot of milk. You want more of such. What do you think you would do?
Interbreed.
Say again?
Interbreed.
Thank you. Can I perhaps cross that cow with a bull and hopefully their children, the baby cows that will be born, hopefully, I'm using the word hopefully, I'll explain why. The babies that will be born hopefully will be able to produce or have a high milk yield as well. So, in the meantime, this is what has happened. You identify the cow among many others. You selected that cow. Are we following? You selected that cow, and what did you do later? You bred that cow, or you breed it. So you've done 2 things: select and breed. Are we clear? This should therefore bring you to one terminology, which is selective breeding. Do you understand now what it means? This is also called artificial selection. The selection of plants and animals by humans based on their desirable characteristics. The characteristics you desired here was high milk yield. Based on that desired characteristic, you selected that cow and then you bred it. That's what our great-great-grandfathers were doing back then, and many of such animals today are found as domestic animals, because when they went into the forest, they saw some animals that were more friendly or less aggressive than others, and they said, I can keep this one around my yard and grow more of it. Then they became domestic animals. Meanwhile, they were all wild before. So based on characteristics, that those animals had characteristics of being tame, not aggressive. They were tame. Okay. So now, I said earlier, hopefully you would have children that can produce high milk yield. Why am I saying hopefully? The reason is because whatever characteristic that the mother had, we are not sure that the offspring will have the same characteristics. Why? First, because the offspring receive only half of the genetic material from that woman, and the other half comes from the bull who doesn't even produce milk. Okay? Secondly, during reproduction, especially sexual reproduction, there is always introduction of genetic variation. So this ensures that the offsprings are not always exactly like the parents. So chances that we have offsprings that produce exactly 60 of high milk yield are not guaranteed. Do you understand? Good. So we are done with this one. Let's go back to our slide here. We identified that the pig there has something that can kill these plants. We will wish that the plant can get that something. But can we interbreed a plant and a pig?
No.
It's not possible. Why is it not possible?
Okay, different species.
Thank you. They're of different species. Do you now realize that the cow here and the bull here were able to interbreed because they were of the same species? Okay? Now, I mentioned the word here, characteristic. We are going to work in terms of characteristics. Do you now realize that whatever this pig is producing, that's something. It means it has the characteristic of doing what? Of producing something. Do you understand? That something is a product of its characteristic. I cannot interbreed plants and pigs because they're of different species. I can't. And nevertheless, I ask myself, do plants have DNA? What do you think?
Yes, they do.
They do. Pigs, do they?
Yes.
Okay, they do. So now, all of them do have DNA. Tell me, the DNA that we find in, okay, the DNA has, what are the building blocks of DNA?
Okay. Phosphate group, the sugar, and the nitrogen bases.
What are the building blocks of DNA? You have gone too far now, more than your shadow.
The nucleotide.
Thank you, nucleotides. What are the subunits of nucleotides? That's when now you start singing that your church song you were singing before. Phosphate, sugar, and nitrogenous base. Okay?
Yes.
Now, the phosphate that you find maybe in the DNA of a mosquito, do you think is the same phosphate in the DNA of mushroom?
Yes.
Huh?
Yes.
Yes or no? I can't hear.
Yes, it is.
Okay. The nitrogenous base you find in the DNA of bacteria, is it the same one you find in the DNA of an elephant?
Yes.
So all these subunits are the same. It means, therefore, that wherever we see it, wherever it occurs, I want you to understand this statement carefully. Wherever it is found, DNA is DNA. Because everything about it is the same across all unrelated species of organisms. Are we clear?
Yes, sir.
So I hope you know the word compatible. Compatibility.
Yes.
Does this mean that all the DNAs around are compatible with one another?
Yes.
Yes, because they are made up of the same components. Are we clear?
Yes.
So they're all compatible. Therefore, this is where genetic engineering comes in. Thanks to genetic engineering, plants, pigs of different species, they cannot interbreed, but what can we do? Can we take, because when I say this pig produces something, that something is most probably a protein. Okay?
Yes.
That something is a protein. And in the body, this protein, to make the protein, where does the information come from?
To make the protein
Yes, to make a protein. Where does the information for making proteins come from?
from our DNA.
Thank you, from DNA, specifically from a gene. Therefore, with my knowledge of little genetics and genetic code, I know that whatever this something is, I might not know it. I'm sure that it is a protein. Secondly, if it is a protein, it means the information for making it comes from DNA. Okay?
Yes.
It comes from a specific section of DNA that we call the gene. That's where it comes from. Therefore, what can I do? From here now, instead of isolating that something, can I rather go and look for the gene in the DNA that has the code for making that protein, which is that something? Can I look for that gene? Is it possible? Yes, it is possible. Okay? If I'm able to find a gene, I find a gene somewhere there, can I take out that gene and put it in this plant? Is it possible?
Yes.
Yes, it is possible. When I put that gene in this plant, will it start functioning? Yes. And when that gene
So
starts functioning, what will it produce? What do you think it will produce?
the same protein that
It will produce that protein that we call what? We call it something. Okay?
can.
At that moment, this tomato plant that you saw before is no longer genetically the same. It's different from the original. Do you understand? So we shall call it a super plant. This is a super plant now. It's producing that something. But that something is a protein. Surely it is toxic to this insect, but not to humans, because nevertheless you eat the pigs and then it doesn't cause you any disease. So I put it in a
Yes.
plant there, it's not going to be toxic there, hopefully. If these insects now feed on this super plant, what will happen to these insects?
It will die.
They will die. Therefore, my super plant here is able to produce that toxic protein as well. And since that plant, based on its genetic makeup now, it is somehow halfway. I want you to understand. It is halfway between the original plant and what it is right now. Are we clear?
Yes.
It's halfway between that original plant and what it is, or it's halfway between the original plant, this is the original plant, and the pig. Because it contains genes from the plants, it also contains genes from where?
Yes.
From the pig. Halfway is like trans or transition. Therefore, This plant is what we'll therefore call a transgenic organism. This is a transgenic organism that we also call a genetically modified organism, GMO. Okay? And these are organisms produced through genetic modification. And what did we do here? We identified the gene. The gene is just a short section of DNA. Let's say this is DNA here. Only part of that DNA, a short section here, could be the gene. What do?
Yes.
I cut it out, I take it out, and then I put here. Are you following?
Yes.
I don't need the whole DNA. The whole DNA itself is very long and it contains a lot of genes that are not necessary for me. All I need is the one that I'm interested in that has the information for making that something. So I cut out, I'll call it my gene of interest. I cut it out and then I insert it there. Cutting and joining. What am I doing? I'm combining, I'm recombining. This aspect of cutting and rejoining is what we call recombinant DNA technology.
Okay.
Is it clear now?
Yes.
So this was just to set the foundation. Now I'll answer your question. I'll go to your question now. Recombinant DNA technology. In this case here, we are going to use it for insulin production. That's what your syllabus deals with. Insulin production. First, why am I producing insulin? I'm producing insulin to help people, diabetics, for example, who are maybe suffering of type 1 diabetes. And type 1 diabetes is as a result of failure of the pancreas to produce insulin. So the pancreas just stops producing insulin. And insulin normally is, in a normal person, is produced on a daily basis. Therefore, if these people can't produce insulin on a daily basis naturally from their pancreas, then they would have to get an external source of insulin. In the most suitable way, it will be an artificial form of insulin produced artificially. In the past, they used to administer porcine insulin from pigs to people. But insulin is a protein, and I want you to remember this. Insulin is a protein because it is a hormone. From your definition of hormones, hormones are organic chemical messengers that are protein in nature and alter the functioning of cells. So all hormones are protein in nature. These are what we call functional proteins. They're like enzymes, they're like liquids. Two major forms of proteins in the body. We have structural proteins. Those are the ones that make up our flesh. The meat is protein. It's semi-solid. Then we have functional proteins. These ones are more or less liquids, like enzymes and hormones. So insulin is one of such proteins, which is a functional protein. Now, being a protein, it also could cause allergies when injected into people because it comes with antigens. Antigens are substances that cause an immune response. It means the moment you introduce them in your body, your immune system is going to react and produce antibodies against them because it sees them as invaders. And when such happens, you might develop allergies. In some people, there were mild allergies. In others, there were severe allergies. In other people, the allergies were even deadly. So people were facing the challenge of either taking the injection of pig or porcine insulin that was extracted from pigs, and dying or not taking it and dying of diabetes. So it was a gamble. You could take it and it's fine with you, it's well compatible, no problem, you are fine. Another person takes it and then it kills the person. But you won't sit down without taking your chances and just die of diabetes. So that was a challenge until when genetic engineering came up and said, why don't we rather cut out and manufacture? I don't want to go to the part of cut out yet. Why don't we manufacture human insulin, which for sure will be compatible with human beings outside the human body? We manufacture it outside the human body and inject it into humans. Is that possible? Yes. So the first question, before we go into artificial production, tell me, by what process do you think insulin is made in the body? Naturally, by what process?
By what process insulin is produced?
Yes, in the body. By what process is insulin produced in the body?
I don't know.
Were you ever taught anything like this, by the way?
No.
Never. We can't force it, okay?
Yes.
So it's correct. Okay. Please, the process is called protein synthesis. And I'm sure you were never taught protein synthesis, isn't it?
Well, the thought is my favorite. Not my favorite. But it was never my favorite.
Huh?
We were taught, but it was never my favorite.
No, it's okay. I told you, did I not say here that insulin is a protein? And I said, please remember it.
You did.
All proteins in the body are made through protein synthesis, my dear. Do you understand now? Or was I speaking
Yes.
Chinese that time? So whatever is a protein, just say protein synthesis. Were you ever taught protein synthesis in your life? Never. That's what you say. Okay? So that's the process by which insulin is made through protein synthesis because it is a protein. And I ask again the question, for making proteins, where does the information come from? You answered it already, from DNA. Therefore, where would it come from, the information for making insulin? It comes from DNA, a gene in DNA. But there's DNA all around our body. If I want to find this information, where do I go to? Do I go to the cells on the tongue? Of course not. I'm giving you already the answers. I'll have to go somewhere. I ask myself, where do I find DNA? I find DNA in several places, one of which in the cell, in the nucleus. So to get DNA, I will have to go to the nucleus of a cell, find the DNA. Am I interested in all of the DNA? No, I'm only interested in a section. That section, we call it what? A gene. And that gene most probably will be my gene for insulin. When I say gene for insulin, it means it has the instructions on how to make insulin. Do I know how to make insulin? I don't know. Do I have the code for making insulin? I don't have. It's just like maybe you are a medical doctor, but you want to open and run a school. Do you have the administrative knowledge of running a school? You don't have. You have knowledge for being a medical doctor. Are you understanding? So what will you do? But nevertheless, you want to do into school business. You don't have the knowledge for that. What do? You have the money, but no knowledge. What do?
Get someone to do it for
You
me.
hire. Thank you. You hire. Are we clear? That's what people do. You hire someone who has the knowledge. It's the same thing I'm going to do here. I don't know how to make insulin. I don't have the code. I don't have the knowledge. What can I do to make insulin? I will hire. Who am I hiring? I'm hiring that gene in the DNA who has the know-how to make insulin, who has the code for making insulin. Do you understand?
Yes.
So that's why I'm going to hire that gene. And next, where do I have the greatest probability of finding such a gene? I won't beat around the bush and look around, everywhere around the body. In my previous knowledge, I know that insulin is produced by what?
Okay.
By the pancreas. It means I'll say, Mr. Pancreas, I know somewhere inside of you there you have the instructions, the information on how to make insulin. Therefore, I'll go and harvest pancreatic cells, knowing that these cells inside of them should have DNA, which has an active gene for making insulin. That's where I'm starting from. Are we clear?
Yes.
Good. So, we are going to get kicked out. Instead of getting kicked out when I'm busy explaining, I'm going to stop the session and then I'll log in. Now. You must log in immediately also.
Okay.
Is it fine?
Yeah.
Sure. I'm not running the full version of Zoom, that's why. Sure. Let me say stop. I was recording.